In this video we will discuss about this MMP9 facilitates N-Cadherin cleavage. So how they check this hypothesis is that they used first this broad-wide MMP9 inhibitor, that is GM, and they also used PMA to overexpress this MMP9. For the control, we can see that when there is no PMA, they are not able to see any MMP9, and when they add this PMA, they are able to see MMP9 expressions. However, when they increase the concentration of GM, they are able to see the simultaneous or corresponding increase in the intact N-Cadherin signals. That is, when there is no GM, there is no intact N-Cadherin. Means all of the N-Cadherins, intact N-Cadherin is being cleaved, and major major cleavage is been done by this MMP9. So when this MMP9 signal goes down because of this GM, there is a rescue of this intact N-Cadherins can be seen. And this thing is true for both the cell lines, that is NPC076, NPC039, and also for this NPC076. That is, with increase in the GM, that is MMP9 inhibitors, they are able to see a decrease in the MMP9 and also increase in the intact N-Cadherin signals. So this indicates that MMP9 somehow facilitates this N-Cadherin cleavage, and they validate this thing using another MMP9 inhibitor, that is MP9-1. In this case also, when they use MP9-1, they are able to see a decrease in the MMP9 formations and the recovery of this intact N-Cadherins compared to the case where they only used PMA. And this is true this is also true for both the cell lines, that is NPC039 and also for this NPC076. Since it is true for two different inhibitors and two different cell lines, this could be they could easily make a point that this is a general phenomena that MMP9 facilitates this N-Cadherin cleavage. And they also shown the quantitative analysis for all the four conditions, that is, in the presence of GM concentration. As GM concentration increase, they are able to see the relative increase in this relative level of intact N-Cadherin increase with increase in the GM concentration. And this is, and also they see the same trend when they used these MPI inhibitors. That is, with MPI inhibitors, the increase in the relative N-Cadherin for both the conditions for both the cell lines is significantly higher compared to the case where they use only PMA. Afterwards, they wanted to check it with another different approach where they used pure activated or inactivated MMP9. So when they used inactivated MMP9, there is not much difference they have seen in the N-Cadherin signals, and also not much difference has been seen in the MMP9 expressions. However, when they used it for this active, when they use this active MMP9, they are able to see a significant decrease in the intact N-Cadherin signals, and also they are for both the cell lines. And these things they have quantified on the right hand side, where you can see that when they use control, when they use only the where they use this control and this pro MMP9, the decrease is not statistically significant. However, when they use the active MMP9 and when they check its signals with control for this relative level of intact N-Cadherin, they are able to see that when in the active MMP9, the intact N-Cadherin signals goes down significantly higher compared to the control case, where they don't use any when they don't treat the cells with any MMP9 or active MMP9. So from these three different approaches, that is two MMP9 inhibitors and one treatment with the pure inactivated and activated MMP9, they are able to see the same trend. So from that, they have made a point that is the MMP9 that facilitates this N-Cadherin cleavage, and this is what they finally come up from the results that are presented in this figure. So if you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.